I want to welcome everyone to this HER2 webinar, and thank you for joining, whether you're joining us live or you're catching the replay after the fact. I'm so excited to be joined today by Dr. John Strickler and advocate Elle Charnisky. I'm going to go quickly because I really want to get to the heart of the matter here. So briefly, if you don't know Global Colon Cancer Association, I'm happy to give you a brief description of who we are and what we do. So Global Colon Cancer Association has 145 members in 60 countries around the world. We are a patient advocacy organization, but because so much of the world, unfortunately, does not have a patient advocacy organization dedicated to colorectal cancer or even all cancers in general, we do a fair amount of direct-to-patient and caregiver outreach, partnering with healthcare providers around the globe. If you are interested, membership is free to patient advocacy organizations. You can join by clicking the link. If somebody could drop that in the chat, you can learn more about who we are. And before I go to the next slide, I just want to say that we know that colorectal cancer doesn't play fair, and that's why we exist. So we want to make sure that where you live in the world is not determining your ability to prevent or survive colorectal cancer. And unfortunately, we know that is the case. Know Your Biomarker is a program that we developed in 2018 and launched in 2019. I'm really proud of this initiative. It's taking a very complicated topic and trying to drill down to what patients really need to know. You can visit knowyourbiomarker.org, and we have a ton of different resources there, everything from questions to ask your doctor, a little bit more of a deep dive. Our treatment impact chart, I think, is incredible because it helps you understand. We also have these tools for providers where you can have in your waiting room these QR codes, because if you know that you have a particular biomarker that you need to focus on, you don't need to, at that moment, learn all the other biomarkers. We kind of talked about biomarkers overall briefly. I have to just start by saying, if you are new to the world of biomarkers, take a deep breath. It's a lot of information, but it is so critical. And sometimes we'll hear from patients a very valid concern of saying, why do I have to learn about this? Because if there was something that was important enough that it could impact my care, it could potentially change my treatment options, or my doctor would tell me. And yes, we agree that in an ideal world, you're going to have that relationship, you're going to have those communications with your provider, shared decision-making. That is the goal. But we know that's really not always the reality. And so, as you'll see and learn in this webinar today, it can be so impactful that even though it's a very complicated topic, and when you're fighting cancer, this is the last thing you should have to do is yourself to figure out what are my treatment options. It's that important. This is not a small quality of life potential change. This could really be groundbreaking for individuals. So I want to just highlight many people that are on the webinar today. You are running patient advocacy organizations. You're walking the journey with patients and trying to navigate the million different things that happen once somebody is told you have cancer. And we know that for many organizations, they tell us this is a lot to take on. And hopefully that's where we can really be a support to you. We want to try to provide you medically accurate information in lay language that you can then share to your patient community. I really want to make sure that we're also hearing feedback. So if any of this material is not resonating, let us know. We want to hear all kinds of feedback so that we can continue to evolve to make sure that Know Your Biomarker is really making an impact in communities around the world. What we're going to focus on today, so we know that biomarkers are really important and that they are a piece of information. I don't want to steal too much from Dr. Strickler's presentation, so I will just share that many of us actually already know a biomarker, even if you don't know, thinking, what is that word? But if you know your weight, if you know your blood pressure, if you know your cholesterol levels, that is technically a biomarker about your health. Now, once we get into cancer biomarkers, it gets a lot more complicated, but it's still with that premise. It's a piece of information about your health, and it's individual to you. So looking at HER2, one comment that I want to make is that HER2 is not the masses of people with colorectal cancer. You'll see, and Dr. Strickler will go over that this is not something that, 9 out of 10, colorectal cancer patients have, nothing like that. But I think this is really how me, from an advocate's perspective, what I've seen is that this is how this cancer is being attacked with smaller biomarkers at a time. And I know many people have heard of MSI high because of what immunotherapy has been able to do for people with MSI high status. I really feel like as many biomarkers as you see up on the screen right now, which is a lot, I actually think it will continue to grow. And so, I think in the cancer space, we often talk about the cure for cancers. And I really think it's the cures. I really see that individual biomarkers play a huge, huge role. And so walking away from the webinar today, I hope that you'll have more information about the HER2 webinar, but that it will also inspire you to engage more on biomarkers in general, if this is your first time engaging. So we want to get to know our attendees a little bit better. So we're going to do 3 rapid fire polls, and we want to get to know who's with us live. So the first question is, how familiar are you with the HER2 biomarker in colorectal cancer? So we'll just take a few seconds to give everybody time to vote. You can lock in your answer. Okay, well, this is quite telling. So many people that are saying that I've heard of it, but I don't know much. That was 44%. And 40% of people are somewhat familiar. So Dr. Strickler, you so that you know where you are, we've got, almost 85% of our audience is somewhat familiar, or they've heard of it, but they don't know much. We have 5% of you are experts that are very familiar, and 12%, this is entirely new to me. All right, next question. All right, poll number 2 is, what is your relationship to colorectal cancer? You can check all that apply. It is multiple choice. We are asking, are you a person diagnosed with colorectal cancer? Are you a caregiver or a family member? Are you a patient advocate? Are you a healthcare professional, or are you other? You can please go ahead and vote now. We'll just give you a few moments to lock in your vote. All right, so looking at our results, we have nearly 20% of the individuals joining today have been diagnosed with colorectal cancer. About 25% are caregivers or family members. 53% identify as patient advocates, 32% healthcare professionals, and other 25%. I would love to know, if you did mark other, if you would be so kind to either drop in the chat what that other role is, or you can message us privately just out of pure curiosity. All right, the last poll question, please. Poll number 3, in what area do you have the most questions about HER2? Now, this is a single choice, so you need to pick which one is of most interest to you. Biomarker testing, treatment options, clinical trials, understanding test results, or living with HER2 positive CRC. All right, wow, well, it's a complete mix, I would say. Treatment options was the leading answer, with 36% of people wanting to understand HER2 treatment options. And then other than that, we're almost across the board, with biomarker testing at 18%, clinical trials at 16%, and understanding test results at 18%, and also living with HER2 positive CRC at 11%. Thank you for answering this poll. That helps us as we're doing this live to make sure we understand who's joining. Elle Charnisky is an advocate that I had the pleasure of knowing for many years now, and she was the patient who really opened my eyes to the fact that people are often afraid of being on clinical trials, but she really shared the level of care that you receive on a clinical trial. You don't have to worry about being a number, because they are certainly paying great attention to you. So I am so grateful for Elle joining. Now, she has done a pre-recorded video, So we are going to go ahead and play that now. And then after that, we will get to Dr. Strickler's presentation. So I'm really happy to introduce you virtually. I'm sure many of you know Elle. She's been an incredible advocate in the colorectal cancer space for many years. So let's play our video now.
My name is Elle Charnisky. I was diagnosed in April 2016 at the age of 36. And I was tested early on for biomarkers and found that I was HER2 positive. At the time, I was told, we're not really going to do anything with this right now, but it could be useful in the future. And sure enough, I got a second opinion at Memorial Sloan Kettering. Dr. Nancy Kemeny had another patient who was HER2 positive and asked her colleagues about it, and was alerted to Dr. John Strickler at Duke, who was heading up the MOUNTAINEER trial. So she, when it looked like we were kind of running out of options because I had failed, I think second and third line treatment. It was irinotecan, I guess is second line. But I had failed that. I had progression on it, so there weren't a whole lot of other options. I contacted Dr. Kemeny and got a call back from her office saying, please contact us immediately. Dr. Kemeny wants to see you. And set up a meeting, and they said, bring the paperwork that shows that I'm HER2 positive. So I did, and we met with her for about 5 minutes, and she said, I'm gonna have you go back out to the waiting room. There's a guy at Duke, Dr. John Strickler, that you really need to see. I think he has exactly what you need. So you'll be hearing from us, and we went out to the waiting room, and they called us back in and said, how do we get you down to Duke? And we were planning to go back home that day. So we had to arrange babysitters and things. But we changed our plans and went to North Carolina instead. And Dr. Strickler basically said, I think things are going to look really different for you, for a while with this. And because I had a very strong HER2 amplification. So the trial was amazing for me. It felt like I was not on treatment. And I got 3 years, of full quality of life out of it. My kids, got their mom back, and it was just amazing. So
that's incredible. So you said that you had the testing, so you had kind of this information in your pocket that you knew that you had the HER2 amplification, but you didn't really know what would come from that. Did you have any comfort knowing that you had this additional information, or what did it kind of feel like to know, okay, I know that I'm HER2 positive, but we're not making any changes now?
Yeah, I think, it was good to hear that it might be useful later on. But of course, when I reached out to Dr. Kemeny, I had already seen her once to get a second opinion. But I had no idea that was going to be the reason she wanted to see me again. So.
Yeah. And then, so once you were in the clinical trial, you said it sounds like it was a really good experience. I'm just thinking a lot of patients are always worried that, clinical trials, you're a guinea pig, or people are afraid they might get a sugar pill, not realizing that placebo is never sugar pill in cancer trials. It would be the standard of care. What was it like for you being on the trial? Did you feel any of those kind of common misconceptions about clinical trials? Did you experience any of those?
No, really not, because, just like I said, Dr. Strickler was very hopeful based on the fact that my amplification was so strong. So, it sounded like it was a great like it was a no-brainer. Um, so yeah.
Sometimes people are worried that on clinical trials, like that they're used to the care they get from their, treating physician and are worried that they might just be a number. Did you ever feel like that, or how did you feel that the level of care you received while you were in the trial?
With it being at Duke, and Dr. Strickler in particular, like, he's amazing. And so, I mean, I have never felt like a number at Duke at all. I will say, like, initially, I had that experience. So, I mean, I can relate to that, but not with the trial.
Do you ever think about how things would have been different if you didn't know your HER2 status, if you had never been tested? Is that something that you've ever thought about?
I mean, I just know I guess I wouldn't, I would have missed out on the trial. And I mean, it's just been a game changer completely for me. If, of course, now I have had to come off of it. I had to come off of it in April 2022. I started it in August 2017. So, I mean, I had almost 5 years of being on it. It was 3 years that I was in complete remission, almost 3 years. But then I was able to have surgeries and stay on for even longer. So, yeah, I mean, I can't imagine if I had not had the biomarker testing, how different things would have gone for me.
And today, how are biomarker test results or your being HER2 positive, how is that impacting treatment options going forward? Do you know?
I did try like some other treatment regimens that, are good for HER2 positive, like in HER2. Unfortunately, though, it's like the amplification isn't there for me anymore. So, I'm just kind of back to like standard treatments at this point. But we're always checking, to see if there are any other actionable targets that we can look at.
What do you say if a patient's kind of like, this is so overwhelming? I'm sure if there's something relevant for me, my doctor will tell me, like, I don't want to research, I don't want to kind of get involved. It's too much to take on. What's your response to that?
I mean, I would definitely say, it's worth having the testing done because it's it's easy. I mean, it's just, a blood test or whatever. Yeah, there's absolutely no reason not to have it done that I can think of.
Great. And I'm just curious, how has sharing your story and being an advocate, how has that experience been for you? Are you naturally somebody who's, very public with things, or how has it been talking about what the cancer journey has been like?
Um, it's been a good experience. I'm not really naturally someone, who's super outgoing with things like that or anything. But yeah, everyone has been extremely accommodating and pleasant, So yeah, I've not had any negative experiences at all.
And what does it feel like when somebody tells you that they got their colonoscopy or that they're following up on symptoms because they have been inspired by your advocacy? What's that like for you?
Oh, I mean, that's wonderful to hear. Yeah, I mean, just to know that it's making any difference at all, in anyone's life. Yeah, it's extremely rewarding.
And it definitely does. I think, um, even we see that it makes a difference. It's obviously not a comfortable topic for a lot of people, which is a shame because, the body is susceptible, all parts of the body are susceptible to cancer. And I think we've heard kind of like famous sayings like you shouldn't die of embarrassment, like that it's worth it to talk about it. So I really thank you for sharing your experience and advocating for others about the importance of clinical trials, of biomarker testing. Well, thank you so much, Ellie, for sharing your inspiring story. I'm particularly moved by the fact that Ellie is keeping it very real, that it's not all sunshine and rainbows, but that knowing her HER2 status, as she said, it was a game changer. And before I turn it over to Dr. Strickler, I think that's one of the really critical points here. You know, colorectal cancer, we often kind of joke we're the underdog. People don't want to talk about it. You know, sadly, research funding is way lower than a lot of other disease states, but the numbers are growing. And I think what's really tough about this disease is that it is so preventable with screening, and it is really tough at diagnosed at later stages. Add to the fact that there are many treatments that have been proven to be safe and effective. And so we have this issue that in countries where they're widely available and largely there are not access issues to these, patients are still not aware, even if they're having the biomarker testing happening, there's a gap and they're not getting access to these treatments. In many parts of the world, it's lagging far behind. And so we have patients who may have these very rare mutations and could benefit from these treatment options. You know, a lot of other diseases, they are looking for screening methods to detect the disease. We have many screening methods for colorectal cancer, or they're looking for treatment options, and we have so many. If you look at combination therapies, we have almost 30 different treatment options for colorectal cancer. Again, with that equity piece being so critical that they are not available in much of the world. And that's where our partnership with advocates like you really comes into play. So again, I thank you, Ellie, so much for sharing your story. We'll share in the follow-up email with the replay, but there's a link to read her story as well on our website. And with that, I will turn it over to Dr. John Strickler, who was already introduced by Ellie as being an incredible healthcare provider who really has had an incredible impact on her life. Dr. Strickler?
Thanks so much. It's an honor to be here today, and I want to thank the Global Colon Cancer Association for giving me the opportunity to be in the same place as people from around the world. It was heartwarming to see where people are connecting from. Some people just up the road from me here in North Carolina in the United States, and some people literally on the other side of the planet. And for me, this is a true honor to be able to be together with all of you today. So we can go forward into this presentation. So the title of today's talk is HER2 and colorectal cancer: what advocates need to know. And I also wanted to give a special shout out to my daughter, Elle Strickler, who's starting her final year of undergraduate education at Swarthmore College, and she has a passion for science and also writing. And I thought this would be a good summer project for her to help me with making these slides today. And she did a wonderful job. So thank you, Ella, for your help with this. So we can go ahead and advance. So here's our agenda today. First, we'll go over broadly what HER2 is. Then we'll talk about who should get tested, how it's tested, and why testing matters. So let's start first with what is HER2? Well, HER2 is a biomarker, and you'll hear this term over and over again. Biomarkers are the foundation of the modern management of all cancer now. And the way I would think about biomarkers is they're like a cancer fingerprint, and they are measurable features of that cancer. And a biomarker can really be practically anything. It can be a protein that's unique to that cancer, or it can be a gene that's unique to that cancer. But the bottom line is we're using that biomarker to help people like me, healthcare providers, understand the cancer and guide a treatment decision. So what is HER2 exactly? HER2 is a protein, and what you can see here on the left is a normal cell, and all normal cells have HER2 on the surface of the cell, and that the purpose of that HER2 is to help regulate cell growth, survival, and repair. So HER2 is a normal part of our body. But on the right, when in a cancer, HER2 becomes what we call dysregulated. So typically due to a genetic event inside the tumor cell, instead of making the right number of HER2 on the cell surface, there's too much HER2 on the cell surface. So when there's too much HER2 on a cell, it turns it into a cancer. It causes that cell to grow uncontrollably, to spread, and and not to die when it needs to die. And so that's kind of the basis of the HER2 positive tumor cell. So why should we test for HER2? Well, as I mentioned, HER2 acts like a switch inside a cancer cell. It tells that cell to grow, to spread, to do everything possible to survive. The good news about HER2, though, is it's druggable, it's targetable. You'll hear the term actionable, and we have therapies that can bind to HER2 and block the HER2 signal. And when we block the HER2 signal, it causes the tumor to either slow its growth or sometimes even die. So when we think about HER2 and colorectal cancer, actually it's taken us many decades to bring HER2 therapies for colorectal cancer. It's HER2 has been known about for years for breast cancer and even stomach cancer. But the reason why it's been very difficult for us to develop therapies for HER2 positive colorectal cancer is that it's relatively uncommon. It's found in on average, about 3% of patients with colorectal cancer. We know that HER2 positive cells behave differently than, say, standard colorectal cancer. It can be a bit more aggressive. It can spread to parts of the body that colorectal cancer typically doesn't go to. So classically, HER2 positive disease can spread to the lungs or even the brain. And also it can respond differently to certain types of treatments. So, for example, it's known to drive resistance to a class of therapies that's pretty standard for patients with colorectal cancer. We call those anti-EGFR therapies. So it has some unique features, and I think that needs to be incorporated into our treatment plan when we know that it's present. So next question is, who should get tested? So if I'm a patient newly diagnosed with colorectal cancer, I think I would want to know my biomarkers. And we know HER2 is important for patients who have either metastatic colorectal cancer that's spread throughout the body, or for patients who have locally advanced colorectal cancer that can't be surgically removed, or for patients for whom colorectal cancer has returned despite our best efforts with surgery. And this, once we know about it, we can incorporate it into our treatment planning. So one of the things you'll hear about quite a bit in this talk and others are the biomarkers that we need to test for colorectal cancer. And I would describe these biomarkers as core to the modern management of most cancers, including colorectal cancer, because it helps us choose the right therapies. There are 5 major biomarkers we need to know to manage colorectal cancer when it's grown or spread, become advanced, or become metastatic. The first biomarker is KRAS, the second is NRAS, the third is MSI, the fourth is BRAF, and then the final one is HER2. Now, there are some other rare targets, but those are the big 5 that we always need to know about. So now an important and very complicated topic. How do we test for HER2? So there's 2 types of specimens we can use to test for HER2. The first is a tissue sample, which requires a biopsy or a surgery to collect that tumor tissue specimen. And this is the way we've been testing for HER2 for literally decades. But the problem with this is it requires an invasive procedure to collect that tissue. So sometimes it's not easy to get that specimen. The other major type of specimen we could use is blood. This is a newer way to test for HER2. It just requires collecting a couple of tubes of blood. This is obviously less invasive. But there are some limitations here. The primary limitation is it can be an expensive way to test for HER2, so this technique may not be available in all countries of the world. Additionally, sometimes there are people who have HER2 positive disease, but the blood test says it does not find the HER2 in the bloodstream. So generally, I would consider tumor tissue to be preferred when it's available. So one of the terms that you'll hear used quite a bit today, both from Ellie and myself and others, is this term HER2 positive. And that could mean one of two things. It could mean that HER2 is either overexpressed or that it's amplified. And let's talk about a little bit what we mean by this, because sometimes we'll use the term overexpressed or amplified and we mean the exact same thing. The bottom line is we have technical criteria for what is a HER2 positive tumor cell. The technique that has been used for literally decades is immunohistochemistry, which we'll shorten up to say IHC. And that's a term that is used to explain the level of expression of the HER2 protein on the cell surface. So the way that works is you take a piece of tumor tissue and you stain that tumor tissue using a protein-specific tag, and you're looking for a color change to occur on those cells so you know, aha, there's lots of HER2 on the cell surface. The other technique that is also old but we'll use in select circumstances is called FISH, or fluorescence in situ hybridization, where you're looking not at the protein, not at the cell surface, but inside the DNA itself, looking for numbers of copies of the HER2 gene. And I'll talk a little bit about how we can use these two old techniques to detect HER2 positive disease. Now, one very confusing point that I need to address is this concept of a HER2 mutation or an ERBB2 mutation. This is not the same as HER2 positive disease. So HER2 positive means that there's too much HER2. A HER2 mutation means that there's the same number of HER2, but the gene sequence has changed in some way. So typically when we talk about HER2 positive disease for colorectal cancer, we're really limiting that to HER2 overexpression or HER2 amplification, not to a HER2 or ERBB2 mutation. We can advance. So there's other ways we can also assess HER2. One modern way we look for HER2, especially for colorectal cancer, is this concept of next generation sequencing. Now, this is not available in all countries. It is a bit more expensive, but the value of this is that you can send one piece of tissue off to a lab and test for all of your biomarkers at the exact same time. Now, the limiting factor here is just the cost of it, but it is a very convenient way to assess for HER2 positive disease at a genetic level. On our next slide, though, there's another way we could do next generation sequence, and this is that blood test I had referred to. And you may hear people talk about this as the so-called liquid biopsy. So instead of sending tumor tissue off, you could just collect a couple of tubes of blood and look for all of your biomarkers at the same time in blood, including HER2 amplification or HER2 fragments. And once again, this is something that we use a lot in some regions of the world, but it can be expensive. Okay. So why might a patient have both tissue and blood testing? Here in the United States, sometimes patients are getting both. The downside is it's expensive to run these tests, but sometimes we like to have the blood test when there's not enough tumor tissue available, or another biopsy isn't possible, or it's not safe, or we need to get an updated profile on that tumor. But the key thing to know is a blood test does not always replace a tissue test, because sometimes something will be present in the tissue that we just don't see inside the bloodstream. Okay, as I mentioned, the oldest way that we test for HER2 is through the so-called IHC test, and this is the most affordable way to test for this biomarker. And as I said, we're using this protein tag to look at the tumor cells, and if those cells change colors, then you've got what's called expression. So if the score is 0, that means the cells did not change color at all. There's no expression, and that is negative for HER2. If the score is 1, that means there's only trace amounts of HER2, and once again, that's negative. Where it gets a little bit more complicated is this score of 2, which is equivocal, meaning we're not sure if there's HER2 overexpression. And there we need to confirm to what's called FISH, which I'll describe in a moment. The highest level of expression is IHC score 3, and that's positive, and that is very actionable and very treatable with some therapies I'll discuss here in a moment. Now, FISH, as I mentioned, has been around for years, and we use this test to actually count the number of HER2 genes present in the tumor cell. And we'll use this test if we're not sure if there's HER2 overexpression. So this is kind of our tiebreaker if we're in that IHC score of 2 category. The final category is NGS, which I just described. And typically on a report, you'll see it listed as HER2 or the gene name ERBB2 amplification. And once again, you can test for that out of blood or tissue. So why does testing matter? Well, HER2 testing can tell us whether HER2 is driving the cancer, whether a HER2 targeted therapy could be an option, or importantly, whether a clinical trial might be available. And there are 3 major classes of therapies now in the clinic that we use to attack HER2. Some of these therapies have been around literally for decades and are off patent now, so are relatively affordable and accessible in all regions of the world. Other therapies are relatively new and on patent and haven't made it to every geography around the world yet. But it's okay. There are therapies that we can use that have been around for decades that are still highly active. The 3 major categories of treatments we have available, and the oldest one we have is the antibody. That's the oldest one. Those attach to HER2 on the cell surface. The second major category are what's called tyrosine kinase inhibitors. These are pills. They block HER2 signaling inside the cell. And then the final newest category are the so-called antibody drug conjugates. And these are basically just fancy antibodies that can deliver potent chemotherapy inside HER2 positive cells. All right, you can advance. So as I mentioned, there are these 3 types of therapies to attack HER2. I mentioned the pills, the tyrosine kinase inhibitors. I've listed some examples of some of these pills here. The oldest one here is lapatinib. That's probably the most accessible around the world since that's off patent. These work by turning off the HER2 signals inside the cell, and these offer the advantage of not only being relatively affordable, they also cross the blood-brain barrier so they can cover brain metastases, but they are daily pills and sometimes they can cause diarrhea. The HER2 antibodies also some of our oldest types of therapies. These are a bit more expensive. There's the oldest one, trastuzumab. There's a newer, what we call a bispecific antibody called zanidatamab that's still only available on trials. These ones are very effective at blocking HER2 signaling on the cell surface. Generally very well tolerated, but they do not cross the blood-brain barrier. They would not attack a brain metastasis, and in rare cases, they can cause toxicity to the heart. The newest strategy is shown here on the far right, the antibody drug conjugates. Some of the examples are shown here. These are FDA approved in the United States for HER2 positive colon cancer. My understanding is they're not approved outside the United States. But once again, this is like precision chemo. So they will basically grab onto the cell surface, get inside the cell, and then deliver very toxic chemotherapy in a very focused way inside the cell. But this is a type of chemotherapy, so it requires close monitoring, and it is associated with some lung toxicity. So last point here, what if HER2 testing is missed? Well, I think it would be a missed opportunity. As a physician, I would lose out on some therapies that I could provide for my patient that might be more effective than currently available therapies. And also, very importantly, there are a bunch of trials going on globally looking at these newer therapies, and this could mean that my patient might miss out on some of those trials. So how can you as an advocate help? I think it's okay to ask your doctor, has my tumor been tested for biomarkers? We know there are some critical biomarkers that help us manage this disease better. It's okay to ask your doctor whether HER2 testing was included. There are a lot of different ways to test for HER2. Some of them are more expensive than others, but there are some fairly affordable ways to test for HER2 if you're in a geography that maybe doesn't have access to some of the more advanced liquid or tissue tests. And then finally, if HER2 is discovered, are there targeted therapies or clinical trials available? These are all very reasonable questions to ask. So just wanted to leave you with a few points today. HER2 is relatively uncommon in colorectal cancer, but it is found in about 3% of patients. When we find it, it tells us a lot about what's driving that cancer, and it helps open up the door to a lot of very effective treatment options. We can test for HER2 in a number of different ways, either by tissue or blood. And more importantly, this just can open up the door for a patient not only living longer, but also living better. And I do encourage advocates to ask about biomarkers and specifically to ask about HER2. And with that, I would like to say thank you to all of you for joining this today. It's been an honor to be here. Once again, want to offer, you know, special thanks to Elle Strickler for her outstanding work putting these slides together. I think she did a wonderful job, and some slide credits are also shown here. Thank you.
Thank you so much, Dr. Strickler, and thank you, Ella Strickler. This was incredible. We are very thankful for you taking a very complicated topic and making such clear slides. So I know there's a lot of questions that are coming through in the chat. So Dr. Strickler, I think you can also see the questions as well, but I will just kick it off and ask. And I think this might be an important thing this just came in 4 minutes ago, and I definitely was making notes that I need to brush up on this, but the difference between HER2 amplification and the different levels of amplification and the HER2 mutation. I know you touched on this, but could you repeat again and maybe explain in a little different way as well, the difference between the amplification, because this is very difficult in not just getting a yes, no. Is that right?
Well, yeah, let's go over this. This is so important and it causes so much confusion, even among doctors, by the way. Okay, so when we say the word amplification, what we're describing is the number of the HER2 genes inside the nucleus of a cell. So we should have 2 copies of HER2. And if it's amplified, we could have 8 or 12 or 16 or 20. And what you heard in Ellie's video is she said, my tumor was very strongly amplified. And what she meant by that is she had a high number of the number of genes was quite high. It was a quantitative number, which told us that this tumor cell was addicted to HER2. It needed HER2 to survive. And when we could starve it of that fuel, then we could really put that cancer into a deep remission. Now, the other term you'll hear sometimes thrown around is mutation. This is not a quantitative change in the number of the genes. There might still just be 2 copies, but they're altered in some way. So the protein itself is folded wrong, or it just creates a functional change in HER2. But this does not have the same importance for us. We don't have treatments for colorectal cancer that are designed for HER2 mutations. So it's a really important distinction between those two.
Wow. Well said. That was a really great explanation, and I really appreciate that. That was explained in a way that I have not heard before, and it makes a lot of sense. There's a question around what HER2 can tell you for prognosis, and
Yes.
asking if that indicates, or does it just guide treatment regimens alone?
This is a topic that I have debated with colleagues around the world for 10, 15 years. We have had very vigorous debates about it, and I would say until very recently, we really haven't had an answer. I personally have had really great success treating HER2 positive disease, and I found that my patients do quite well. So I've always thought that it's not that poorly prognostic, but there have been some really interesting studies done globally that have found that actually when you look at a HER2 positive patient and compare it to a person with the same profile who isn't HER2 positive, that the HER2 positive patients appear to have more aggressive disease and tend to have worse survival. I think the key thing to know is if you treat the HER2 positive disease, you can actually cancel out the negative prognostic impact of the HER2 positivity. So, this was actually shown in breast cancer many years ago, where HER2 positive disease was a very concerning, worrisome change. And then once all those new HER2 therapies came in, it wasn't so poorly prognostic anymore. And I think that same phenomenon has happened with colorectal cancer as well.
Great, thank you. We have a question around Lynch syndrome,
Yeah.
and if there's a correlation or increased risk for CRC syndrome survivors.
For So is the question whether is that specific to HER2
I think so,
or
yeah, for her
yeah.
too. So is there an increased risk?
So here's the really interesting thing about Lynch syndrome, which is um, nearly always with colorectal cancer, microsatellite instability high. Not always, but nearly always. The interesting thing about Lynch syndrome is that is a condition where people are born with a DNA repair defect, and it causes a type of mutation in a cell that causes that protein to fold in different ways. It almost never causes amplification of the gene. So, in some way, I don't think in my entire career of treating HER2 positive disease, I've ever seen somebody with Lynch syndrome having HER2 positive disease. However, I have had many patients with Lynch syndrome have
Hmm,
HER2 mutations.
interesting.
And we talked about just
Yeah.
a moment ago how HER2 mutations, we don't have great drugs for that yet for colorectal cancer. I hope that will change soon. But right now, HER2 mutations would be common in Lynch syndrome, but are not targetable.
Okay. And now we have some questions around the testing, the frequency. So would you get retested for HER2? Are there guidelines on how often you should be tested?
Yes, great question. And this answers actually many questions that I'm seeing in the Q&A. So obviously we want to test when somebody's diagnosed and find that HER2 positive disease. I really like to test the metastatic lesion, not the primary tumor, because the metastatic lesion is kind of that dominant clone that we're trying to attack, right? We want to attack the cancer that's spreading through the body. And then you treat that cancer with some kind of anti-HER2 therapy. It could be tucatinib trastuzumab, could be lapatinib trastuzumab, could be tucatinib deruxtecan, which goes by the trade name of Enhertu. But what happens is when you give that patient an anti-HER2 therapy, that cancer is also under a lot of pressure. And so you're killing off all the sensitive clones. You're killing off all the HER2 positive disease. And then eventually, in many cases, that patient develops resistant disease. And we want to know when that happens, what happened to that tumor? How did it figure out a way to evade our most effective treatments? And so we'll either do a new biopsy with a tumor tissue biopsy, or we'll do a blood sample. And you heard Ellie mention that in her video that she ran some blood tests. And that was a liquid biopsy to kind of sample what happened to her tumor tissue. Sometimes when the patient progresses on an anti-HER2 therapy, it's because they lose the HER2 biomarker. And that's what Ellie had mentioned in her video. And there, then we know we should not give more anti-HER2 therapies. Sometimes, however, HER2 is kept and the HER2 is still as strong as ever, but that cancer cell has acquired some other new feature that allows it to resist the attack. Sometimes that new feature would make it sensitive to trastuzumab deruxtecan or Enhertu, or sometimes it might make it sensitive to some other new type of targeted therapy. So yes, to Olga's question, yes, you can develop progression on an anti-HER2 therapy and still overexpress HER2. And that's classically the patient that we would want to give Enhertu to or trastuzumab deruxtecan. That person who's had maybe a tyrosine kinase inhibitor and an antibody still is overexpressing but is resistant, that would be one where we want to give that new class of therapy, an antibody drug conjugate.
Got it. So if a patient does have biomarker testing, HER2 is tested, and nothing pops on the results, can they confidently say that they know in the future, or could it also change the other way? Could the HER2 show up later on in the disease if it didn't previously?
Yes, this is possible.
Okay.
That HER2 could show up later. So the scenario where we would be most likely to see that would be somebody who has what's called KRAS and NRAS wild type or non-mutated disease, microsatellite stable, gets a lot of anti-EGFR therapies, and then over time, maybe over a few years, would develop we call it a clone. A cancer cell would develop HER2 positivity and use that to escape the anti-EGFR therapy, and that would open that patient up to that type of treatment.
Okay, really helpful to know, because I think sometimes we're searching for, okay, can I check this off my list? But what I'm hearing from you is that, continued conversations, it's not a guarantee that just because you didn't have it. There's a
Right.
lot of questions coming in through whether chat or the Q&A around promising clinical trials. And I'm just wondering if you can speak to that a little bit. And then Tom specifically asked, if does research for HER2 benefit both breast and colorectal cancer in the same way, a pan-cancer therapy between two tumor types? But definitely there's a bunch of questions asking about what's out there in clinical trials that we should be aware of.
Yeah, one of the things let's start with the first question. Can research in one disease help another? I would say that patients with HER2 positive colorectal cancer can thank those patients with breast cancer that signed up for these trials 20 years ago, because it really opened up the target for all these diseases. We learned, hey, we're helping all these patients with breast cancer, but wait a second, there's this small group of patients with colorectal cancer that have the exact same gene alteration. And by the way, those treatments that work so well for HER2 positive breast cancer work just as well for HER2 positive colorectal cancer. So yes, we are seeing that this rising tide, raises all the ships. And now there are some biological differences between colorectal cancer and breast cancer. They're not perfectly, the lessons don't always transfer perfectly, but we have benefited greatly from the drug development in this space. The next question is regarding clinical trials and what's going on in the space. There is a major global international phase 3 trial ongoing right now to bring one of our most effective anti-HER2 treatments, tucatinib and trastuzumab, up into the first line setting for patients with HER2 positive colorectal cancer. That is hopefully going to be finished soon, but if you're in a country where that trial is ongoing and it's a newly diagnosed colorectal cancer, please find your nearest clinical trial site and try to enroll in that trial, because if we have a positive phase 3 trial there, I think it will further expand the access globally for this important therapy.
Great, thank you. And just for clarification, could it be somebody who is not newly diagnosed but is new to the metastatic setting?
Yes.
Okay, so we're
If
not
they
only
have,
talking about people that are not engaged because they are like just diagnosed. They could be a member of our patient communities for a long time,
yes,
but
the
not
key thing
metastatic.
is, yeah, not metastatic yet. So it's what we call first line treatment of metastatic disease, maybe meaning they have never had treatment for disease that has spread through the body. So that would be the trial we have. But I will also point out there are some other very intriguing trials going on globally looking at newer anti-HER2 therapies. It is really hard to find those trials. Um, Certain parts of the world have access to these trials, other parts don't, and that's a frustrating thing for all of us. But but as we develop these drugs, I'm hoping they'll become more and more accessible throughout the world.
Wonderful. Well, we are almost to the end of time. So I will say that if we didn't get a chance to answer your question, we'll connect with Dr. Strickler and see about maybe releasing a blog with some of the highlights here, and we'll make sure to get you an answer. I just want to thank you so much, Dr. Strickler. This was incredible. I've learned so much. I think that you shared information in a very approachable way. I know that many of us who do not have a science or medical background, it can be quite overwhelming, but I really do thank everybody for engaging. We do have a resource that's on our website, on our HER2 page. Once we have this recording, we will also host this recording for people to watch on our Know Your Biomarker microsite on the HER2 page. So this is the guide, what advocates need to know. Also, the call to action. I really appreciated the very reasonable questions that Dr. Strickler posed that we can ask our providers. The more that we're talking about this disease, the more that we're talking about biomarkers and encouraging providers that we do want to have these conversations, the better it is. I do want to thank Pfizer for their support. Their support allowed us to host this webinar and create some important materials. So thank you very much. A quick call to action that when you log off of this webinar, you will be getting a very brief survey. And we really appreciate if you can share your feedback. We really believe that with the biomarker conversation, we're going to be going through biomarkers one at a time. So if there's a biomarker that you would like to have us cover, please let us know. And thank you so much for joining us. We really appreciate all of you around the world. Take care. Thank you.